Hello everyone. This lecture is about the process of recombinant DNA technology. There are different steps are involved in this process. We discuss all these steps clearly and in brief. So here it starts with first, in the process of recombinant DNA technology, first we are going to isolate the desired DNA, our gene of interest, which we are going to produce a product. Second, it is involved in later on the fragmentation of the DNA by using restriction endonucleases. Next, separation and isolation of our desired fragment from the entire DNA fragments by process called gel electrophoresis. Later, the gene is amplified by process called polymerase chain reaction or PCR. Later, so our gene is introduced into vector and resulting in the formation of chimeric DNA or recombinant DNA. Then uptake of this recombinant DNA by the host cell. Then later on the culturing of this host cell with recombinant DNA into bioreactors. And finally in the downstreaming process our gene product is extracted. So these are the 8 steps involved in the entire process of recombinant DNA technology. So we discuss all this. First it starts with isolation of donor DNA. Fine. DNA. So source of DNA. First from the cells we have to extract the DNA. Say for example, if we are extracting the DNA from plant cell, animal cell, bacterial or fungal, first cells must be treated. We have to open the cells and extract our DNA. So like for example, if it is plant cell, we are using an enzyme called cellulase to break open the cells. That is cell wall. In case of animal cells, lipase and protease. Later on in the fungi, it is called chitinase. For bacteria, we are using lysozyme enzyme. By using these enzymes, we are going to break open the cells. And this entire cell products along with the nuclear membrane will be lysed. Then later, so this is treated with different enzymes which can digest proteins, carbohydrates and lipids, RNAs except DNA. So all these cell fragments of the cell, which includes our DNA also, is now sent to ultracentrifugation, and after that we get a suspension, and in the suspension our DNA is present. So how do we extract our DNA? So this DNA can be separated by treating this with chilled ethanol. So addition of chilled ethanol to the sample resulting in separation of our DNA fragments. So finally we get all the DNA fragments. So these DNA fragments, that means this entire DNA can be seen as a layer between buffer solution and the ethanol. Then by using a glass rod, this is by using a glass rod or by a spatula, we can separate out DNA strands which can be seen as fine white threads. So fine. Now we have obtained our DNA. So that is our donor DNA. Now that DNA contains our gene of interest. So next step, how do we separate it? The second step is fragmentation of DNA at specific locations by using restriction enzymes. So this entire DNA is subjected to restriction endonucleases. So this restriction endonuclease will keep the DNA into different fragments. So like see here, there is a fragment 1, this is fragment 2, fragment 3, fragment 4, fragment 5. Like this, it is divided into different fragments. Among these fragments is present our desired gene. So we have to now separate these DNA fragments by a process which is known as gel electrophoresis. Fine. So here we are going to take agarose gel. That means this is gel plate. So this gel plate is formed by using agarose gel. So agarose gel is extracted from seaweeds, and this gel plate is placed on the casting tray, and then it is given a charge. One end of the gel plate is negatively charged and other end of the gel plate is positively charged. So here there are wells. Wells are for loading DNA samples. So here there are several wells are shown. So here you can see 1, 2, 3, 4 wells are shown, because this gel electrophoresis can be used when we are comparing the DNAs of different people. Different source of the DNA for comparing, we can use different wells for placing or for loading different strands of the DNA. Here, because we have only one kind of DNA, we can just place them in one well so that the DNA strands or DNA fragments can start moving from one end to the other end. So all the DNA fragments are loaded at the negative terminal. As we all know that the DNA strands are negatively charged because of the phosphate. So when the power is switched on, the DNA fragments will start moving from negative to positive terminal. Always smaller the size of the fragment and more faster and more speed the distance what it travels. So always the smaller fragments can travel very faster. Larger fragments will keep moving very slowly. So now you can see that all these DNA fragments, like smaller the fragments, they can keep on moving slow. So as a result, all these fragments of the DNA are separated on the gel plate. So among these fragments, our desired DNA is also present. Now on the gel plate, DNA fragments cannot be seen. So as a result, we are going to treat this with a compound called ethidium bromide. So this ethidium bromide, under exposure to UV light, it gives bright orange colored fragments. Now these fragments can be compared with the standard DNA ladder. So if we know the length of our DNA fragment, our gene of interest, our desired gene, then with the DNA ladder we can compare the size and we can extract it. And what is the process called? That process is called as elution. So fine. So now we have separated our DNA fragments, and among these DNA fragments we have isolated our gene of interest. Next thing is, when we get our gene of interest, we get very less number of copies, because with a few copies or a limited number of copies, we cannot continue the process. It's difficult to introduce into the host cells. As a result, we have to amplify our gene of interest, because if we have more number of copies, then the process becomes very simple. So as a result, for amplification of a gene of interest, we use a technique which is called polymerase chain reaction. In this polymerase chain reaction, we are using our desired DNA, and along with that we are using, the most interesting thing is here, enzyme. That enzyme is Taq polymerase enzyme. So the link for this PCR is provided, so you can just go through that lecture. So here in the entire process of PCR, there are 3 steps are involved. So the first step is denaturation. So what are we going to do in denaturation? In denaturation, first, in the PCR machine, the temperature is raised to nearly 94 degrees. At this temperature, the hydrogen bonds break. Am I right? So the 2 strands of the DNA separate. The 2 strands, the double-stranded DNA gets separated into 2 strands. This is denaturation, first step. This is followed by the second step, which is known as annealing. In annealing, the temperature of the reaction mixture drops down to around 50 to 60 degrees. At this temperature, the primers will go and bind. This is primer binding, which is called hybridization. So primers bind at the 3' end of the template DNA strand. Just you can find out here. Next, this is followed by the last step, which is called chain extension, or I can say that it is primer extension. Once again, the temperature is raised to around 72 degrees. At this temperature, the Taq polymerase keep on adding nucleotides to the primer strand, and the primer strand grow towards the 5' end. So this is how the DNA strands can be multiplied. Like this, we can multiply DNA into 2 to the power n number of molecules. N number of molecules. 2 to the power n number of molecules. That means after 30 cycles, we can make into 1 billion copies. So this is what amplification of gene of interest using PCR. Fine. Now we have amplified our gene of interest, our desired gene. Next, which is followed by ligation of our DNA fragments into a vector. So fine. Most commonly, we use vectors like plasmids. So imagine this is a plasmid. This is a plasmid vector which we are taking for this process. So already we have cleaved our gene of interest by using a restriction enzyme. Now, important point is the restriction enzyme which is used for cleaving our gene of interest, right, on the donor DNA. The same restriction enzyme sequence must be present on the vector also, so that both sticky ends of the donor DNA as well as vector DNA will join. So this is easiest. So remember, the restriction enzyme which we are using to cleave our gene of interest, the same restriction enzyme also, the sequence must be present on the plasmid. That is a vector. So fine, we are using the same enzyme and we are going to cleave the vector. Fine. After this, our gene of interest is added, and this is with an enzyme called ligase. All the sticky ends are connected by enzyme ligase. So it results in formation of our recombinant DNA, which is also known as chimeric DNA. Recombinant DNA can also be called as chimeric DNA. So now we have introduced our gene of interest into a vector, which is here. I am showing you the plasmid. So now this is called formation of chimeric vector. So fine. What is the next step? Now this chimeric vector or recombinant DNA must be introduced into host organism. So there are various different hosts we have. Like, and we can introduce this into bacterial cells. We can introduce this into plant cells. We can introduce this directly into animal cells. We have different type of host cells according to our requirement. So imagine here, suppose we are making insulin. That is the only example what everyone will give it. So here, for example, insulin. Insulin is a human gene. We have extracted insulin gene, and later on this insulin gene will be introduced into a vector called PBR322, and that must be introduced into a host called bacteria, for example. So DNA fragment or DNA molecule is negatively charged. I said it is called as hydrophilic. So as a result, the DNA molecule is hydrophilic. So the cells cannot accept it directly. As a result, so these DNA fragments must be treated with divalent cations like calcium. So whenever we treat with divalent cations like calcium, then the cells become competent. So this is a normal bacterial cell. This normal bacterial cells, we are first making it into competent. That means we are treating this with calcium chloride. That is calcium divalent cation. Then its membranes becomes permeable. Then it is given shock treatment, or we call it as heat shock, where this competent bacteria is first placed in ice cold medium. From there, it is transferred to a temperature of 42 degrees. Once again, it is sent back to ice cold medium. By transferring in this way, the bacterial membranes becomes permeable for taking our gene of interest, that is vector. Now here, after making it, the bacterial cells, the membranes becomes permeable and are recombinant DNA. So this is a recombinant DNA. that is plasmid, that is vector with rDNA, is introduced into cell. Now such process is called transformation, and this is called transformed cell. This is how we introduce into bacteria. Like we have different methods. Like we can use, in case of plant cells, we can introduce by using TI plasmids. Suppose if I wanted to introduce recombinant DNA into back plants, we can use TI plasmids. And we also follow one more technique which is called gene gun method. Gene gun method or biolistics. By using this method, we can introduce our desired gene into plant cells. Whereas when you talk about animal cells, in the animal cells, this desired gene can be introduced directly by micro injection into cells. Directly to the nucleus of the cells by micro injection we can introduce. Or there is one more technique which is called as retrovirus mediated transfer, that is called transduction. So by using all these methods, we can introduce our desired gene into host cells. So here I said, imagine this is insulin gene, we are introducing it and we are making it. We are making the gene product. In such cases, we are using here bacteria. It is called E. coli bacteria because PBr322 plasmid, okay, will have an ORI site of E. coli. So as a result, we have to introduce it. Fine. After introducing this into bacteria, this is called a transformed cell. Now with the help of genetic markers like ampicillin or tetracycline, these are the two antibiotic resistant genes are located on the PBr322. So by placing them in the antibiotic resistant medium, we can find out our, transformed cell with the recombinant. So once we find out our transformed cell with the recombinant DNA, we are going to separate them. Next, last, we have to obtain our gene product by using bioreactors. Fine. Now we have first isolated our gene of interest, and then we have separated our gene of interest. We have multiplied our gene of interest, and then we made a recombinant DNA. We have introduced it into vector, and finally these vectors are introduced into host organism. Now in the host organism, we have to clone it, the gene. And also we have to get our gene product. Say for example, insulin gene I am introducing into E. coli. So E. coli will express, those are called expressing host. There are hosts, some are cloning host and some are expressing host. So we are introducing the gene in such a way that the host cell can even produce our gene product, express our gene product. So fine, how do we do this? So for this we are using bioreactors. So listen, bioreactors are steel tanks. Bioreactors are steel tanks which are measuring around 100 to 1000 liters. Now what is the specialty of these bioreactors? Because when we are culturing or we are going to manufacture our gene product in the industrial way, so there must be some conditions we need to follow. Under controlled conditions, we need to maintain a constant pH. We need to maintain optimal temperatures, and for all this we require a bioreactor. So in the bioreactors, all these conditions can be under controlled conditions we can maintain and we can provide. There must be proper aeration in the bioreactors. So we use two types of bioreactors. One is called stirred type and second is called sparge type. So here I am explaining you only stirred type of bioreactor. Sparge is similar to that. Okay, but the difference I will tell you what is the difference between these two. Now in these cultures, there will be a continuous culture as well as batch culture. So in the continuous culture, what happens is we keep on adding our the nutrient medium and also our starting material, and we every time keep on obtaining our product. That is continuous, which is also called open system. And in batch fermentation, we completely load the bioreactor with a nutrient and as well as our starting material. And then once if the entire product is formed, the product is completely removed and the tanks are cleaned, and once again we go for a next batch. So this is called batch fermenter. It is called as closed system. So remember, bioreactors are of two types, open as well as closed. Open is continuous culture, closed is a batch fermentation. So fine. So here we are using a stirred as well as sparger type. So let us just have the setup, look at setup of this stirred type of bioreactor. So guys, this is a stirred type of bioreactor. Here, you just observe this bioreactor. It is the base of this is curved base. Just you can see this is curved base. Now this curved base is for uniform aeration. Thorough mixing. That's the reason why it's curved base. Next, here is a mortar. A mortar is fit. This is a mortar. A mortar is fit. So this mortar, a shaft is arranged to the mortar. This is shaft. Shaft is arranged to the mortar. So there are different fans you can see in this. These fans have different functions. Like the fans which are present here. I'm just marking these fans as 1, 2, 3 fans. These fans, when this motor is switched on, the fans will keep on moving. So they will provide uniform aeration. Whereas the fan which is located on the top, these blades, are acting as foam breaker. Foam breaker. That means what all the foam that is formed during the process of fermentation, that is for breaking that foam, we use this fan present on the top. Next, in this entire reactor, nutrient is added. This all yellow one is the nutrient. The entire tank is filled with 2/3 of nutrient. Now, you can see there is a pH scale to record the pH. What is a pH? Whether it is acidic or basic, that is important. And here there is a thermometer. So thermometer is to record the temperature. So fine. Now, once this is completely loaded with the nutrient, then the final requirement is inoculum. So you know what is this inoculum? Inoculum is our starting material. So what is the starting material? Our E. coli cells, transformant cells with our recombinant DNA. This is recombinant DNA. E. coli with recombinant. This is our starting material called inoculum. Now this inoculum is added. Fine. So this process of fermentation inside the reactor keeps working, and the E. coli keeps on multiplying, as well as our gene product is also produced. Our gene is expressed. Then first with the pH scale, we are going to measure the pH every time. If suppose the container inside the reactor, if it is highly acidic, then we are going to add base, alkali. If it is highly alkali, then we have to add acid. So because we have to maintain a constant pH. Fine. Here is a water jacket. So this entire reactor is surrounded by a water jacket. This is a water jacket. So what is the advantage of this water jacket? So if inside the reactor the temperature is very high, we are going to circulate the cold water. Inside the reactor, if the temperature is very, very less, we are going to circulate hot water, because we need to maintain uniform temperature inside the reactor. Fine. So if this entire process keeps working inside the bioreactor, then finally we are going to extract our product through this outlet. So what's our product? Our product is now containing our gene as well as the bacterial cells, everything we have in that. Fine. Next, what is the next step? Next step is downstreaming process. That is, what are the content that we have obtained finally? The content which we have obtained, so we have to extract that. Okay, so actually downstreaming process processing is the name given to the stage after fermentation. That means after the process of fermentation, or you call it as biosynthetic stage. After biosynthetic stage, the entire processing is called downstreaming. So when the desired product is recovered and it is purified. So our container, that means our bioreactor contains our gene product, microorganisms and everything. So this downstreaming process involves few events. So this includes first separation. So we get two products. One is the solid and the liquid product. One is the solid and second is liquid. Fine. So this solid product do not contain our gene product because it contains the cells, fragments and everything. So we are going to separate this from our liquid. This liquid portion contains our gene fragment. So remember, for different gene products, downstreaming process is different. We go for sublimation, okay, and we go for different techniques to separate our gene product. So then after this separation of the gene product, then we are going to purify our gene. Suppose if it is insulin, we are going to purify it from our liquid part. We get only insulin. Then we have to find out whether this insulin is really working or not. This is testing of the gene product. After testing, so we test this on different animals and finally a group of humans under experimental condition, we check this. If the gene is working, then it is going to be packed and marketed. So students, this is how the gene, the desired gene product is finally prepared by using recombinant DNA technology.